Welcome to the 27th Annual Benjamin Ide Wheeler Society Tea. First, we start with a lecture, and normally I introduce our tea members. I'll do that at the tea afterwards because I really want to save as much time as we can for our really distinguished speaker. I mean, every year we bring you distinguished speakers, but I've never had an opportunity to introduce a Nobel Prize winner, which I'm going to do right now. So Professor Randy Schekman is Professor of Cell and Development Biology in the Department of Molecular and Cell Biology and also a Howard Hughes Medical Institute investigator. Randy Schekman shared the 2013 Nobel Prize in Physiology or Medicine for his role in mapping out the machinery by which yeast cells sort, package, and deliver proteins. This is shortly after he received his prize from King Gustav of Sweden. And this is where he received another valuable prize, a lifetime parking pass. And this is where he received the University Medal at UCLA from another king, the king of basketball, Kareem Abdul-Jabbar. And he can probably tell us a little bit more about how that came. So Professor Schekman's research on yeast, diabetics worldwide use insulin produced and secreted by yeast, and most hepatitis B vaccines around the world are also secreted by yeast. And his research now is focused on the process of membrane assembly, vesicular transport, and membrane fusion among organelles of the secretory pathway. I don't know what any of that means, and I'm sure he's going to explain. So it's my deep honor and privilege to introduce Professor Randy Schekman.
Great to be here this afternoon and to share a little bit of what I've been up to for the last 50 years or so. I thought I'd take a step back as I describe the work that we've done here at Berkeley for the last 38 years by telling you a little bit about the influences in my life that led me in the direction that I've taken in science. I grew up in Southern California. My dad was an engineer, my mom was a homemaker. I became interested at a very early age in microorganisms. This started with a toy microscope that I got in junior high school. And I don't know why, but I picked up a sample of pond scum, and I was amazed to see the life forms that swim around in pond scum. You wouldn't believe that the diversity of life at this microscopic level is as rich as what we see in our daily lives. And that just fascinated me. Well, I did some science fair projects, and that's how I really got into science. But the key moment came when my father at the dinner table told me that, well, after all, it was really just a toy that I was playing with and wasn't really a serious instrument. And I kind of got offended by that remark, and so I started to save all the money that I was making babysitting and mowing lawns to raise $100 to buy a professional microscope. Now, I kept the money in an envelope in my closet, but I found as time went on that the money kept disappearing, and it turned out my mother was borrowing my money to pay for groceries. And I was so offended one afternoon after finishing a lawn that I bicycled over to a police station, and I told the police officer that I was running away from home because my mother was stealing my money and I couldn't get my microscope. So my dad was called in. He wasn't too happy. But that afternoon, I got this microscope. A Bosch & Lomb microscope that was my prized possession throughout the rest of high school. Here was one of the science fair projects that I performed in 9th grade, and this was my big moment throughout the school year. I was a pretty good student, but I didn't get all A's because I was entirely focused on science and math. I then had the opportunity to go to UCLA, and that picture that you saw of my hero, who was then called Lou Alcindor, he had just been recruited to UCLA when I decided to go there for college, and I waited up all night the fall season of 1966, waited up all night outside of Pauley Pavilion I paid $6.25 for an entire season's of tickets. 25 cents a ticket to see him, and that he then came out to this graduation ceremony at UCLA a few weeks ago to give me the UCLA medal was quite a treat. Well, at UCLA, I had the opportunity as a freshman to learn under a Nobel laureate by the name of Willard Libby. In my the third term of my freshman year, he taught an honors section of chemistry. He had obtained his PhD here at Cal, but he taught at UCLA for many years. He was the inventor of carbon-14 dating of ancient materials, so really stupendous achievement. But the major important thing of that class for me was every student in that section was assigned to a chemistry professor's laboratory to work under his or her tutelage. And I was assigned to a lab of someone who'd gotten his PhD here at Cal also, and he taught nucleic acid biology and biochemistry. And the most important thing I learned from him was when he gave me a copy of a book written by a fellow you may have heard of, James Watson, the fellow who discovered the structure of DNA. Well, his first textbook was called The Molecular Biology of the Gene. And I read it as though it was the Bible. It was just a revelation to me. And I decided from that exposure just in my freshman year that I was going to disappoint my parents and not go to medical school. After all, I was going to be a scientist because that was clearly what was my passion. During the years that I was at UCLA, I worked in a laboratory studying chromosome replication, how viruses duplicate their DNA. And so that was a very important experience for me. I guess I didn't have that in here, but because it taught me how to look at problems at the microscopic level, how to isolate molecules and to take them apart and to study how they work. And after my experience as an undergraduate at UCLA and then as a graduate student at Stanford under the direction of another laureate by the name of Arthur Kornberg, a man who was arguably the greatest biochemist of the second half of the 20th century, I decided that I wanted to focus on a problem that was rather different, but one that was important in a basic understanding of how cells work and how they produce molecules that are shipped outside of the cell. Let's have a look at a cell that you may not have seen, but I know you all know about. This is a cell in your pancreas called the beta cell. It's the place where all of the insulin gets made for your body. Insulin in these cells is housed in granules. These are little packets that themselves are surrounded by a biological membrane that is roughly the same as the membrane that surrounds the entire cell. So this cell is devoted day and night to the production of insulin that will be discharged when these granules come up to the perimeter of the cell and they kind of pop by a process called membrane fusion, discharging their content to the cell exterior. And then it goes into your bloodstream and it's delivered to peripheral tissues where it allows cells in your body to take up glucose that you've generated by digesting food from a meal. So this is a really fascinating process because it explains how molecules that are made inside the cell, protein molecules, can end up outside of the cell. Normally, the cell is contained by a membrane that surrounds the entire compartment, and yet there are literally thousands of different protein molecules that manage to escape the barrier that surrounds a cell. So cells manufacture over 20,000 different kinds of protein molecules that do all the things that make life, the rich chemistry of life. But some of those have to be exported, shipped outside of the cell. Molecules like insulin, antibodies, all the proteins in your blood, these have to be manufactured inside of a cell and then shipped outside of a cell. So the question is, how does that happen? Well, here's the membrane that surrounds not only the outside of the cell, but the membrane that surrounds the capsules inside of the cell that that package proteins like insulin to be shipped outside. The membrane consists of a couple of different kinds of molecules. These red stringy molecules are lipids. They're the kind of fatty component of a membrane, and they are organized in two layers called a leaflet or a bilayer, and that provides the kind of insulation that surrounds a cell and keeps molecules inside of a cell or keeps other ones outside of a cell. Now, membranes are distinguished mainly by the presence of these kinds of protein molecules, these green blobs. These are protein molecules that are embedded in the membrane and give it its characteristic identity. So your red blood cell will have proteins that are different from the cells of your liver or of or nerve cells. And so these proteins are functionally the identity of each cell and of the capsules inside of cells. Well, this man was a real pioneer in understanding how the membranes inside of cells are organized. His name is George Palade. He did his pioneering work at Rockefeller University, where he developed a way of looking at cells in the pancreas, in the mammalian pancreas, not only the beta cell, but the cells of the pancreas called the exocrine cells that are producing enzymes that go into your digestive tract. And he devised a way of looking at the complex membranes in a cell and determining how they are related to each other. So this process of packaging a molecule of proteins into a capsule and getting it out of the cell requires a kind of a conveyor belt-like process where proteins are manufactured in the cell and then conveyed from station to station within the cell until they're eventually encapsulated to be discharged to the outside of the cell. Anyway, he was a brilliant man. He won the Nobel Prize in 1974, and just after returning from Stockholm, he came to San Diego, where I was then a postdoctoral fellow at UCSD, and gave a lecture at an important meeting of the American Society for Cell Biology, and it was really fixed in my mind the approach that he took in trying to understand how these molecules could progress within a cell, and it gave me some insights into what I might want to do in my own career, and which I have done here at Berkeley. Well, let's have a look at the various membranes that Palade was able to see inside of pancreatic cells. One of his principal achievements was to use this instrument that you saw behind him. It's called an electron microscope. It focuses an intense beam of electrons on a piece of tissue, and wherever there are dark staining things in the specimen, they bounce electrons off, and you see a negative image then on a screen that's projected below the electron beam. So you see where the electrons have penetrated the sample as light, and you see where they have not penetrated, or they bounced off as dark. So it's a kind of an impression of a cell. He also found ways of taking tissue and preserving it so that it wasn't destroyed by the electron beam. You have to be very careful in preparing a tissue. It has to be chemically fixed and embedded in a plastic resin, and then you use a very sharp diamond knife to make thin sliver slices through the cell, so thin that you can actually see inside of a cell. And that's the image you see here that was one that he took many decades ago. It shows an elaborate network of membranes inside of a cell of the pancreas or possibly of the liver. It's a network of membranes that has this funny name. It's called the endoplasmic reticulum. Now, some of you may remember this back from high school biology or college biology. It's a structure that is very involved, has a large surface area inside the cell. It's basically a large template. And on this template, Palade discovered these dots, these dark staining dots. These are things that bounce electrons off, and so it shows up dark in the beam that goes through the tissue sample. These dots, he demonstrated, are the places in a cell that manufacture protein molecules. It's a kind of a little machine. It's a really amazing machine that stitches amino acids one next to another, sometimes making stretches of amino acids that extend for thousands of residues. And that is how protein molecules are made. They're stitched together, one amino acid after another, like a sewing machine producing, weaving an elaborate cloth. In this case, it's a protein molecule. When the long stretch of amino acids finally emerges from the ribosome, the intrinsic chemical nature of the amino acids dictates how this string folds up into a compact shape that creates a molecule, a protein, possibly an enzyme that has its unique role in the cell in catalyzing other chemical reactions. Well, in the pancreas, almost all of the proteins that are made in the pancreas are going to end up being shipped outside of the cell. And what Palade discovered is that these dots, these ribosomes, thread the proteins that are going to be made and secreted from the cell into this clear space called the lumen in the interior of this ER membrane. So that seems odd. Why would you do that? Why would you stick something inside of a membrane, inside of a cell, when what you really want to do is to get it out of the cell? Well, turns out that this pathway, this first step, is involved not only in producing proteins that get encapsulated for secretion outside of the cell, but it's also involved in making all of the different membranes inside of a cell. So this first station is a station that involves the assembly of lots of different proteins that are going to go to different places in a cell, secretion being only one of them. Well, the next organelle that Palade figured out its function is an organelle that was first discovered by an Italian cytologist by the name of Camillo Golgi. It's a structure that takes his name. It's called the Golgi apparatus. This was an organelle that was kind of obscure in its function for many decades. Golgi won the Nobel Prize. He won one of the first Nobel Prizes in physiology medicine in the first decade of the 20th century for this and other discoveries. But it remained a kind of an obscure discovery found in all cells until Palade in the 1960s and 70s figured out that it's the next station in this assembly line process where proteins are first put into this thing called the endoplasmic reticulum, or ER, and then they're shipped to this next station. It's like being shipped from one station to another in a subway system, and so proteins are collected here and then distributed to various destinations in the cell. Well, finally, Palade was able to see in greater detail what happens at the very end. This capsule that contains, in this case, insulin, moves up and touches the perimeter of the cell. These two membranes, the membrane that surrounds the capsule and the membrane that surrounds the cell, literally touch each other, and at a crucial point that's very tightly regulated, the two membranes merge. They recombine, a process called membrane fusion, the result of which is that now the single membrane exposes this granule to the outside of the cell. So it goes from being inside of the cell to being shot out of the cell. Now the granule dissolves after it leaves the cell, and the proteins that are part of this granule diffuse away eventually into the bloodstream, where insulin then can be circulated to the entire body. Well, so we have snapshots then of these three principal subway stops along this pathway, and what Palade did, that I won't go into any particular detail, was to deduce with really brilliant experiments, technically very challenging experiments for the 1960s, that these three steps are related to one another temporally. And indeed, he further extended by analogy to cells that aren't actually exporting proteins like insulin. Even nerve cells use this process. So here is a section cut right through the middle of a nerve terminal. This is the nerve ending. In this case, it's a nerve ending that is directly adjacent to a muscle cell. It's called the neuromuscular junction. So nerve cells get activated, and when they're activated, at the very end of the process, there's a cluster of these little capsules that are touching the surface membrane of the cell, and at the right moment, when there's an evoked potential on the membrane, these little capsules merge by that process of membrane fusion, and the interior gets discharged into the gap, which is called a synapse, between the nerve cell and the muscle cell, or between two nerve cells, and neurotransmitter chemicals then spill out, touch the muscle cell, and activates, let's say, a muscle contraction event. So this process, where capsules are carrying neurotransmitters, turns out is almost identical to the very last step, where a molecule like insulin is secreted from a pancreatic cell. And in this case, you know that this process is crucial for many things that we do. The neurotransmitters include molecules like dopamine, which is deficient in patients with Parkinson's disease, and which then is crucial for controlling movement, or molecules like serotonin that control mood, or molecules like acetylcholine, which is crucial for the communication between nerve cells in the brain, and which is destroyed progressively in patients with Alzheimer's disease, who lose large regions of the brain and thus destroying the cells that are so busy churning out and secreting acetylcholine. Well, so here's the path. It really should be called the Pilati pathway. He was able to show in experiments that molecules start off in this structure. They move in little small carriers to this next station, demonstrating that the Golgi apparatus really has an important role. And finally, they become collected into these granules, which move up to the cell perimeter, and then by process of merging or fusion, shoot their content to the outside of the cell. So this is what I learned in 1974, when Pilati had just returned from Stockholm to talk at this meeting. And I was trained as a biochemist, this fellow Kornberg at Stanford, and I looked at this. It was thrilling to see in its contour, but to a biochemist interested in how things really work, how these machines work at a molecular level, it was in a way unsatisfying. I really wanted to understand at a deeper level how this process could be organized, and that meant finding out what the chemical catalysts were that allowed each step in this process to proceed. That is, how these proteins could be threaded through a membrane in this structure, how little vesicles could form and move and deliver their content by merging with the next membrane. What are the protein molecules that organize and control this whole process? And could one then delve into this at a mechanistic level and uncover really fundamental processes in cells? And eventually, with the hope, of course, that by understanding this process at a molecular level, that you would have more insight into the molecular nature of diseases that affect this process. And there are many such diseases. So armed with that insight, or that hunch, let's say, I moved to Berkeley. There's a fellow many of you know by the name of Dan Koshland, a wonderful figure here in the life sciences. He was the chairman of the biochemistry department when I was hired, and he was really my second mentor after Kornberg, and taught me really how to live a life in science, and I really greatly appreciate what he did for me. So here it is, Berkeley, 1976. I arrived and decided that I would do something rather different than what anyone had done at the time. Instead of working on animals and isolating tissues, things like the pancreas, I decided to work on yeast cells, on baker's yeast, because baker's yeast was already clearly known to be like an animal cell in its internal architecture and in the genes that it has to organize its growth and division. But it really wasn't understood with any detail about how yeast cells manufacture protein molecules that are going to be exported. And yet there were powerful genetic and biochemical tools available to understand this basic process in yeast, much more sophisticated than was then available to study these processes in human cells or in animal cells. So here's yeast. This is a clump of yeast cells. This might be an image that you would see if you looked at with a high-power microscope at the surface of a grape. All the cells in the population are uniform. They're genetically identical. That's a really big advantage. They grow, of course, differently than our cells do. They form a bud. And this bud, you can see another one here, grows during the 90 minutes to 2 hours or so of the growth cycle of a yeast cell. And as it's growing, it's producing enzymes that go out into a wall that surrounds the cell and allows the yeast cell to scavenge sugars that are manufactured by the grape, and then use those sugars to create glucose that allows the yeast cell to grow and divide and ultimately to ferment to make alcohol. So it's a really useful organism. It's simple to grow, and it makes quite a nice preparation for biochemical analysis. Here's an image now looking inside of a yeast cell, really much less dramatic than what you saw in those pancreatic cells that I've shown. Nonetheless, there are things that look a little bit like the process in a human cell. There are membranes that are endoplasmic reticulum-like. There's a mitochondria, and this is the energy forming factory in a cell that makes the currency of life, the energy currency of life, ATP. There are even things that might be Golgi membranes, and it's particularly useful when you cut a section and it has a nice G there. That really helps you identify what it is. Now, we were particularly interested in little carriers, little packets that are congregating under the bud portion of the cell. So this is the part of the cell that's growing during those 90-minute, 2 hours of yeast cell, the cell division cycle. And it was known even before 1976 that these little packets carry enzymes that go into the cell wall. You see this cell wall around here. But we imagined, and me being one of my first graduate students, Peter Novick, shown here, working in what is now Barker Hall, looking very much like a 1970s kind of guy with the long hair. I similarly had long hair, but I'm proud to say that I still have more hair now than Peter does. But Peter was a fantastic, this is what made being at Berkeley so great. Peter was essentially my first graduate student, and he was just brilliant, technically brilliant, had great ideas, golden hands. He has since gone on to his own career, first at Yale, now at UC San Diego, and just 2 months ago, he himself was inducted into the National Academy of Sciences. So he was a star from beginning, and he still is. And so what Peter and I did was, going back to these images, we had the idea that these carriers, these little packets, might not simply be producing enzymes that go into the cell wall, but that the membrane that surrounds these packets would become the building block of the cell surface. So when the packet or capsule fuses with the membrane that surrounds the cell, its internal content would go out, but that the membrane of the vesicle would then allow the perimeter of the cell to enlarge. So if you make that guess, if that's the hunch, then of course the prediction is that if you disrupt a gene that's required to manufacture these capsules or to deliver them by fusion to the cell surface, that should kill the cell. And there's a way of dealing with mutations in genes that kill cells. I mean, normally if you just would delete such a gene, the cell would die and you wouldn't have anything to work with. But there's a way that geneticists have developed over many decades that allows you to study genes like that are crucial, that are absolutely essential. And that is by introducing a mutation into the gene that cripples the protein but doesn't kill it. And you can cripple a protein in a way that allows it to kind of work at room temperature, but in some cases, the crippled protein simply unfolds and is no longer active when you warm the cell up to human body temperature. So it's a mutation that's called a temperature-sensitive mutation. It produces a protein that gets along at room temperature but can't hack it when the cell is stressed by exposing it to a temperature that a normal cell would be happy at, 37 degrees centigrade. Temperature-sensitive, lethal mutations. Now, of course, taking a step back, we were, after all, proposing to kill yeast cells. And this being Berkeley, you know, such thoughts of killing things are taken very seriously. And so we had to, propose the work to an experimental subject committee, and there were protests outside of my laboratory. End the torture in the labs. Yeast have feelings too. But, you think about it, every time you bake a loaf of bread, you're killing 1 trillion yeast cells. So we weren't alone in this torture. So fortunately, we were able to convince the committee, and obviously I'm kidding, that yeast are not sentient beings, and we went on our merry way. Well, so within a few months, Novick had a really nice way of making yeast colonies that were temperature sensitive for growth, but you don't know why. There are 1,500 different genes in the yeast genome that can be mutated in that way, and not all of them are involved in protein secretion. So we had a way of screening through mutants to find ones that conform to our predictions. And the prediction was that enzymes that would normally end up outside of the cell in such a mutant would instead build up inside of the cell. And that had never been seen before, but we're kind of confident that we could find that. And so he found such a mutant, and then, just by chance, this wonderful man, George Pilati, came to give 2 special lectures at Berkeley. And at a lunch meeting, Novick told him about his results, and George said, wow, that's really interesting, but now you want to look by using the electron microscope at a slice through the cell to figure out what it looks like. And that was one of the great moments of my career, came when Novick called me excitedly from the electron microscope room in the basement of the building, and I was up on the 6th floor, and I ran down, and I saw this image, or something like it. So recall the image I showed you a moment ago where you see just a few of these little capsules inside of the bud portion of the cell. But in this mutant, when you warm it up under conditions where it's going to die, but not yet, it starts to manufacture these vesicles, but they can't go to where they want to go. They can't fuse at the cell surface. So instead, the cell piles up with thousands of these vesicles. And if you cool the cells back to room temperature, they'll survive if you haven't kept them at the high temperature for too long. And these things will re-engage the pathway, and secretion is restored to the cell surface. Well, that was very exciting, and we published these results in 1979 in a journal called The Proceedings of the National Academy of Sciences, a journal that Koshland was the editor of, and I, many years later, served as the editor of. And this was a paper that crucially was communicated by Dan for publication. Well, this was exciting, and it gave us the clear evidence that we were on the right trail. And so what Novick did over the next 18 months was he repeated this procedure over and over and over again with refinements to isolate many more such mutants. And a year and a half later, we were able to publish another paper that demonstrated that a couple of dozen new genes emerged from our analysis, where each gene played a different role in this assembly line process. Many of the genes act at this very same late step, implying that there are many different protein molecules that must work together to make this last step possible. In fact, we now know that the very first gene that Novick identified is found, in fact, all of these genes are found in the genomes of all higher organisms. In fact, this first gene, which encodes a protein that we call SEC1, SEC short for secretion, is in all of your nerve cells. It's right there at the synapse, organizing the activity of those little capsules that are delivering neurotransmitters outside of the nerve terminal. And it is crucial in doing that in all organisms and in all cells that secrete anything. Well, there were other kinds of mutants that turned up in this amazing collection of Novick's. Here's a mutant that produces a structure that really hadn't been seen in anything like this in a yeast cell, but it clearly is a Golgi apparatus. In this mutant, the subway station stops midway through and things just pile up in this organelle. And the organelle itself builds up to much higher levels than you normally see in a yeast cell, at least. And finally, another group of mutants block this process at the very first step. And in this mutant, this structure, the ER, builds up to much higher levels than you normally see. It's a very extensive network within the cells. Well, using other techniques, very simple techniques, we could then line up all of these different classes into a pathway. Here is the pathway. It looks very much like the events that you saw in an earlier slide that Palade had deduced, but in this case, crucially, each of these steps is defined by a number of different genes. They have these funny designations, but in every case, every single number means that there's a gene, and for every gene, that means there must be a protein molecule that acts at a different stage in this pathway. So this was a big development in the field because it allowed one now to really go in and study molecules and mechanisms, the dream that I had when I started this effort. Now, around this time that we made these observations, the biotech industry was growing up in the Bay Area, and a local company started by Bill Rudder and Ed Penhote called Chiron took an interest in trying to manufacture useful quantities of human proteins like insulin. And it was clear from our work that yeast cells could manufacture their molecules by the same process that human cells use. And so it was not a big leap of imagination to predict that you could take a yeast cell and transplant the recently cloned gene for human insulin in a yeast cell, provision it with signals that would allow it to be manufactured in a yeast cell, and then it would piggyback, be hijacked, and secreted outside of the cell. That worked after a few years of engineering. And now we know that it's a very efficient way of manufacturing recombinant human insulin, so much so that 1/3 of the world's supply of recombinant human insulin is made in yeast cells, piggybacking along this secretory pathway. At the same time, Chiron found that they were able to use yeast to make what is now the exclusive supply of hepatitis B vaccine for the entire world. In the early 1980s, it was possible to be immunized against hepatitis B. At that time, patients who were infected with hepatitis donated their blood, and it was possible to isolate particles from the blood of infected patients, special particles that had the shell of the virus but lacked the infectious core nucleic acid of the virus. And that material could be purified, injected into a patient who would then develop a good immune response but not become infected. Well, unfortunately, within a few years after the early 1980s, the very same patients who were donors of blood were also becoming infected with HIV. And no one was willing to isolate these particles from the blood of patients who were also infected with HIV. And so what Chiron did was they took the protein of the shell of the virus, and just like as I've described with insulin, transplanted it into a yeast cell, and lo and behold, the yeast cell started to build up little virus-like particles that filled up the entire cytoplasmic volume, and it's these particles that can now be isolated. And if any of you have been immunized in the last 30 years for hepatitis, you've had these little particles manufactured in yeast cells. Well, okay, so now we're in around the mid-1980s. We'd made these discoveries. They were clearly, in my mind, just laying the groundwork for what I really wanted to do, which was to get in and to understand the molecules themselves that operate this process. And one way to do that was to take a lesson from the master, my graduate mentor, Arthur Kornberg, and break the cell open and try to understand how this process works by manipulating the proteins and membranes in a broken cell preparation. That is a classical way of discovering a biochemical process. You break the cell open, you spill out its contents, and you probe with various tools the molecules in this crude extract, and ultimately you try to purify them, put them together in pure form in the test tube, and see how they work. Well, we did that, and I'll give you just one quick example of a successful approach. We were able, within a few years, using the discoveries that we made earlier, to develop in the test tube the ability to manufacture these little containers, these little capsules or vesicles. And when we manufactured them in the test tube, we found that they were surrounded by a kind of a fuzzy coat. You can see that here. It doesn't look very well defined, but I'll show you an image that shows that it really is a very regular structure. So each little capsule, as it forms in the cell, is literally enveloped by a kind of an exoskeleton structure in the cytoplasm of the cell that pinches this membrane, in this case from the ER, to make a little freely contained capsule. Well, in discovering these genes and in discovering how these vesicles are formed, we learned about 10 years ago that the genes that are required for this process in yeast also turn up as defective in certain patients with congenital defects in very specific aspects of secretion. So I'm just going to tell you a quick story of one example. We, about 10 years ago, were alerted to a Bedouin family in Saudi Arabia that had an unusual craniofacial disorder. So these kids develop cataracts at a young age. They have facial dysmorphism. The soft spot, fontanelle on the top of their head never closes. And it turns out it's because of a defect in bone growth. And bone growth is intimately connected to the production of things like collagen and connective tissue that allow the bone to grow and, in the case of the soft spot, to enclose. And these kids are specifically defective in one of the proteins that we discovered in yeast. And we were able to use our tools, our biochemical tools, to figure out at an atomic level exactly what was going wrong in these kids. Not that we could offer them much help, but we could at least understand in some detail how it worked. Now here's a snapshot that was really quite surprising to me. This is a sample of a slice through a normal skin cell, a normal human skin cell. And this structure is the ER that you've seen before. This is a slice through one of the parents of the kids. These parents are carriers, heterozygous. They're perfectly normal. They have no disease. And the ER is more or less normal. It's not too bad. But here's a slice through the skin cells taken from one of the kids. And look at this. This is an amazing distortion of this ER structure. Really very extreme, more so even than the original yeast mutants that we found in this gene. And we see all this granular staining material inside in the interior, which we've shown is in fact collagen. And we now know that these cells have a real problem manufacturing and exporting collagen, which is so important in connective tissue, and which almost certainly is at least partly the basis of the disease. Well, there are many examples. I won't tell you all of them. We found mutations that affect the production of lipoprotein particles. We found mutations that affect brain development, all in the very core elements that we discovered in yeast. And that leads us to a conclusion that I want to emphasize, and that is the unity of biology. When something was invented or evolved in a microorganism billions of years ago that solves a basic problem, how do you ship things outside of a cell? You see that process is pretty complicated. It makes good sense that you would want to use that process over and over again as organisms evolve. And we see evidence of this in this process and in all processes. An amazing conservation of these genes. Evolution has borrowed pieces and used them over and over again. Well, this all culminated last year in Stockholm. I was awakened on the morning of October 7th at 1:20 in the morning. I'd just come back from Germany where I'd received some other award, so I was tired, and fortunately I went to sleep early that night. I woke up, my wife heard the phone ring. She yelled out, "This is it, this is it." We had no advance warning, I can assure you. And I stumbled to the phone. I heard this very comforting Swedish voice, and my first words, "Oh my God, oh my God." Because I knew that my life would change from that moment on. So it's been a blur since then. This was certainly a highlight. But I want to return to where we started, and that whatever happened to that microscope? So remember, there was my prized possession all those years in high school. Well, you know, I went away to UCLA, and they had their own microscopes. I didn't need to bring mine along. I kind of forgot about it, but I brought it up to our home here in El Cerrito when we moved 38 years ago, and it sat around collecting dust. But then I got a call from the Nobel Museum shortly after the prize was announced, and they asked if I had any artifacts from my past that I'd like to share. So I went down to our wine room, I picked out the microscope, and if you ever go to Stockholm, you can visit my microscope in the museum. Complete with the story about how I had to run away from home to get the microscope. Thank you for your attention.
Thank you, Professor Dr. Nobel laureate Schekman. We have time for a few questions, and a couple of our team members from Gift Planning have some microphones. If anybody has a question.
Well, a friend of mine recently died, so I'm asking this question. Can you illuminate us more about cancerous cells? He died of cancer.
Yeah. Well, let me emphasize again, I work on how basic science, but I feel firmly that when you understand something at a fundamental level about how a cell works, that will apply to disease. And so let me tell you a cancer story that follows from just what I said. Really quite unexpected. This is why I feel so strongly about basic science. So I told you how the local company Chiron used yeast to make insulin, and they used it to make these hepatitis particles that are the source of immunization for the disease. Well, it turns out that liver cancer is 90% attributable to infection by hepatitis B. As a patient progresses with the disease, eventually the liver is destroyed, and these patients become subject to liver cancer, 90%. So now we have an effective vaccination, and as long as people have that vaccination, we can reduce by tenfold the incidence of liver cancer. So, and there are so many stories like that, and I feel that's why our efforts in institutions like Berkeley are best spent focusing on basic processes, basic science.
Any other questions?
There's a question. Oh.
Did the three of you work together or totally separately?
Yeah, so I shared the prize with a fellow named James Rothman at Yale and Thomas Sudhoff at Stanford. We know each other. I've known Rothman since the very beginning. I was his host in 1977 when he interviewed for a faculty position here. Probably better off that he went to Stanford, but because we collaborated and were competitors for some years. But then around 1994, we shared a prize, and we're fortunate to share a number of prizes, and at that point we became good friends. So I've worked with him, I've published with him. Sudhoff I've known, but I haven't worked with directly.
Well, as everyone feels, huge thanks for a truly fascinating story. It was a wonderful personal as well as scientific story. And I'm going to ask a very mundane question, which is we've heard a lot within our universities about the need to partner with for-profit companies, big pharma, an obvious one. And I wonder, you've had that firsthand experience, how it worked, and if you have thoughts about it.
Well, I think it's great when we have the opportunity to work along with companies because they have very different goals, they have very different ways of pursuing things. And my relationship with Chiron was wonderful. I worked as a consultant for them for 20 years. I was able to watch as they, engineered the secretion of insulin in yeast, a project that I wouldn't have done in my own laboratory, or manufactured hepatitis B, or discovered, in fact, a new virus called hepatitis C. That was all done in those laboratories. And so that relationship was, I think, a very productive one, certainly satisfying to me. And I think many of my colleagues engage in those kinds of collaborations. At another level, some of my colleagues have actually developed things in their laboratory here that have had direct commercial application. And I'll give you a spectacular example. A former colleague here by the name of James Allison, an immunologist, was working on a basic problem of how T cells are entrained to attack other cells that the body wishes to get rid of. So T cells are killer cells that bind to and can kill, in principle, cancer cells. But animals also have proteins in the blood that kind of regulate that process, that kind of dampen that process and keep T cells from attacking our own cells. So Allison had a good idea. He said, well, there's one particular molecule that puts a brake on this process. It's called CTLA4. What if I temporarily inactivate that protein? Would a T cell be unleashed to attack a tumor cell more aggressively than it would otherwise do in a normal situation? So inactivate a breaker, unleashing T cells to kill tumors more effectively. He did this in the animal. It worked swimmingly well, and over a period of years, he developed this into a commercial product, collaborating directly with first biotech companies and then with big pharma. And 3 years ago, the world was exposed to the first fairly effective treatment of melanoma. This treatment, which you may know about, is one of the first and most promising forms of immunotherapy against cancer. Fully 20%, and it's getting better, but at the time, 20% of patients treated with this immunotherapy go into essentially permanent remission. This had never been seen before for melanoma. There's now another breaker molecule that's been identified, and if you attack both breakers at the same time, I just heard from Allison a few months ago that success rate is up to 80%. So we have the prospect of a cure for melanoma. Indeed, this kind of immunotherapy may actually be useful for many other forms of cancer. So these are all the result of an intimate relationship between what we do at a very basic level in the university and the commercial sector. I think that is a very healthy kind of relationship. What I'm less eager with is when the federal government, and particularly the NIH, tells us that we have to be directly in the business of drug discovery. I'm not really qualified to do drug screening, drug discovery. I'm in here to understand how cells work. I think that when I discover something of a basic nature, it's better to be applied in the private sector. So I would favor more basic science in the university setting, especially at a place like Berkeley.
We have time for one more.
Professor, beside the remarkable story of science you shared with us, I think there's another story that you could be telling, one that should appear in the Atlantic Monthly or the New York Times or maybe on 60 Minutes, about the connection between great teaching and the results that you've generated. Starting at UCLA, going to Stanford, ending up with Dan Koshland, there's a tremendous story about the value of higher education in America and the support it needs from the public. Maybe that's another paper you can be publishing.
Yeah. Well, so I'll thank you for that question. I obviously agree with you. I feel so strongly about it that I donated my prize money to Cal. I did so to honor my mother and sister, who both died of cancer. So we now have chair in their name. And I'm pleased to say that with the efforts of my colleagues and in university relations, we are now almost up to the $2 million level that we need for that chair. But if that's just one small story, the need is much greater.
Thank you, Professor Schekman.
Sure.